Hi, my name's Cosmo. I'm going to tell you about a cool project I've been working on for the last month or so and that I'm submitting for this hackathon called Quarantine at Home. And it's based around a computational drug discovery technique that's being leveraged against the proteins that have been determined from the novel coronavirus, COVID-19. So, about a month ago, maybe a month and a half ago, there was a lab that released the three-dimensional protein structure of the spike glycoprotein from the coronavirus attached to the human ACE2 gene right here. So this is the receptor inside the human cells that the virus spike protein recognizes, and they have the full three-dimensional protein crystal model of those two proteins bound to each other. And you can see here that there's a certain contact patch that occurs between the virus protein and the human protein. And the crazy idea that I had was maybe I can find small molecule drugs that get in there and bind to the virus protein a lot stronger than this human receptor would. And if I did that, maybe I could find a drug that would disrupt this binding. So I went and started playing with software called Autodoc again. I learned how to use Autodoc in my PhD many years ago, and I realized that I could use Autodoc to draw a box around the spike protein and then find small molecule drugs that bind to it. Now, to do that, I need a big database of drugs, and luckily at my university, UCSF, there's a lab that has a database called Zinc that has over 1 million different virtual drugs that are available for download in all different kinds of categories. Drugs that have been tested in humans, drugs that haven't been tested in humans, and there's a huge diversity of drugs that can be downloaded, effectively just three-dimensional coordinate files of all the atoms, and they can be placed inside the Autodoc software, and you can determine if these drugs bind to the protein and with what energy and binding strength that they bind. When I realized this though, 1 million drugs, that's a lot of compute power. So about a month ago, I started working on this project where I would use the central server to coordinate work, and I would let volunteers from around the world download a client software, kind of like the original SETI at home, but I'm calling it Quarantine at Home. And what this software does is if you run it on your computer with either Docker or what's coming soon is a Windows version, you can let your computer download small molecule drugs from the UCSF database and dock them against various proteins found in the coronavirus. And then once those jobs are done, it sends the result up to the server. And here you can see the most recent jobs that have been run on the platform that have a really good binding energy targeting the various receptors of the coronavirus that we're targeting. So we're targeting the spike protein of the virus that binds to the human receptors, and we're targeting the protease of the coronavirus, this gene right here. And with every single job result, you get exactly what drug molecule did the trick from the database. And you also get the ability to click on this results link and see what the binding patterns looked like of that given drug, like what shapes that drug would take when it bound to the protein surface. So what we're doing next is we're going to make, as I said, the Windows version of this, which I think will open up the door to hopefully at least several thousand people running this on their Windows computers. Right now, Linux computers, Linux users that have Docker installed can run either a CPU or a GPU version of the software and join today. Right now, we've got about 10 volunteers running the software, and we've run already 40,000 jobs over the last 2 weeks, and that's an incredible large amount of work that we've done already. Anyways, let me know if you have any questions.